Hello everyone, welcome back to our lecture series. Today we'll be looking at chapter 5. In this chapter we'll be looking at what I usually tell my students is called biochemical instrumentation. That is the way I tend to look at this chapter. This is biochemical. That is what basically we're looking at here. So anyway, but the topic according to the book we're using is just called protein purification and characterization techniques. That is exactly what we're going to be looking at. So we're going to take a snapshot of those methods and techniques used in purifying, extracting protein, purifying protein, and then characterizing them. And let's take it off from there. Isolation of protein from cells. Of course, proteins are biomolecules. Biomolecule means that are found in living organisms. That's what we call them, biomolecules. Now, these proteins are found inside the cell. Of course, we remember in chapter 1, we took a snapshot of what the cell looks like. A typical animal cell, of course, we have a nucleus at the center of it, and then it's going to have the liquid part, the cytoplasm. It's going to have the cell membrane, and then it's going to have the bean-shaped mitochondria, and it's going to have other stuff, like the ribosomes and the endoplasmic reticulum, and all the rest of them. So what simply means you're going to get these proteins out of this cell. You have proteins inside the cells and in a mixture of cells. That's where you get the proteins from. So you're going to get these proteins from inside and around these cells. So there are many methods you can use in doing this. So many techniques are performed to eliminate the contaminants. Now, if you want a particular specific protein, assuming you want to isolate a particular protein, let's say the protein you want to isolate is lactate dehydrogenase. Now, lactate dehydrogenase is an enzyme. Of course, it's a protein as well, LDH. You just want this particular protein. What it means is that this protein is existing. If it's existing in the cytoplasm, it's in a mixture of other proteins and other stuff. So you want to get it out of that particular system and get it in a pure form. All right? So many techniques are performed to eliminate those contaminants that you don't want and arrive at a pure sample of that particular protein of interest. So before purification begins, protein must be released from the cells. That's what you need to do. Some of these proteins are intimately associated with other parts of the cells, with the organelles. The organelles are those smaller other parts of the cells. They are either localized in one subcellular organelle or the other. A very good example is that the lactate dehydrogenase can be found in the muscle cell. Of course, if you want to get an enzyme such as pyruvate dehydrogenase, it is found in the mitochondria. Now, what it means is that you're going to break down the cell, you're going to break the mitochondria before you get to your pyruvate dehydrogenase. We call it PDH. Because pyruvate dehydrogenase is the enzyme that breaks what? That converts pyruvate to acetate-CoA before it enters the tricarboxylic acid cycle. So this is exactly what happens. Now, so this is a protein purification scheme. Look at what happens here. In a protein purification scheme, two things are going to, there are important things important columns that we're going to see very interesting changes in. But before we do that, now you get a crude extract. You usually have it in more volumes. Of course, the moment you begin to do extra purification steps, the volume begins to decrease, which makes sense. If you're going to get any raw sample, it will be big. By the time you start extracting, the volume will begin to decrease. So by the time you get into more sophisticated or more detailed specific steps and purification steps, you find out that the volume begins to decrease. Now, this is proportional what happens at the total amount of protein you start with. You see, you have the more amount here, you extract some. So extraction means you're leaving just a small part of a whole. And if you keep going down, it's going to decrease. Now, the same thing happens with the total activity. The total activity of that particular protein or enzyme is the overall, I would say, the total activity measures the overall activity, the overall performance of that protein in that cell. Now, remember, that is not what you're looking for. You're looking for the specific function of that protein. That means you're going to isolate it away from other guys that help it to function. So that means that as you're moving through these purification steps, the total activity decreases again because you're removing like the coenzymes, you're removing other subcellular organelles where it is localized until it stays alone. However, what increases? The specific function of that protein, not the way it functions with other protein components, but individually begins to increase. Look at it here. If you have a specific component, the more you are purifying, the specific activity of that protein begins to increase because the specific function in what you're looking for begins to increase. And again, if you look at the percentage recovery, you find out that as you go down these purification steps, the percentage recovery begins to decrease. You look at all these steps, that every other one decreases as you go down the purification step, except what? The specific activity, which is exactly what you're looking for when you are purifying a protein. You want to look at its specific activity and what it can do. So that will take us to those steps one by one. The first step you want to do in getting at a protein is homogenization. Homogenization simply involves the process of doing what? Breaking the cells open. You see, these cells are localized. These proteins are localized inside the cells. So what it means is that you need to find a suitable way to break them open and get these enzymes out. Now, the method you're going to adopt in breaking them open shouldn't be so harsh that you're going to even destroy the protein or the enzyme of interest you're looking for. So the techniques you usually hear is that you grind the tissue in a blender. You can use a blender to break the tissues into small parts, or what you can say is to increase the surface area. And then you can use what we call the Porter-Evengem homogenizer. Sorry. You see, the Porter-Evengem homogenizer is this now. It consists of a tube and then a thick rubber, which you use in pounding the cell until you break them. So you use the T-wall tube to, pound those cells until you break them. And when you break the cells up, what happens? The organelles gets out immediately. You can also use sonification. Sonification simply means using very high sound waves to break those cells open. All right. So the next step you're going to do is, after you break them open now, right? So inside these cells, you have the organelles. Remember, if you break a cell open, you have the organelles, you have the enzyme, you have everything mixed together. But there's a need for you to begin to separate these things. The first thing you're going to do is to use the centrifuge. The centrifuge machine, of course, works on the basis of what? The centrifugal force or the centripetal force. Now, so this process, you have the rupture cells are centrifuged using several increasing gravity over time. So what it means is you're going to increase the speed of the spin. A centrifuge spins round. If it's the one that goes clockwise, no, that doesn't look good. Let me make it beautiful. Now, it spins this way. So assuming it is spinning this way, you want to keep increasing the speed. So if you start with a lower speed, the next one, you're going to recover some, you're going to increase the speed again. So look at what happens in this step. What happens in this step is that now, after breaking down, this is homogenization. After breaking it down with your Porter-Evengem homogenizer, now you transfer this into a tube. Now these tubes are now doing what? You now move them into this tube. These tubes are fed into the centrifuge machine. Remember, anytime you're going to put a centrifuge, you must balance it two centrifuges. So if you put a tube here, okay, let me give you an example. If you put a tube here to spin, you have to balance it with another tube. If not, that one is going to fall off. So you have to balance them with an empty tube, or you can just add water inside it and make as if it has something in it. So the first centrifugation is done at 600 grams for 10 minutes. When you do that, what do you see? You find that you get you're going to get a clear layer. The clear layer is called the supernatant. The lower layer is the unbroken cells, the residue. So after the first spin at 600 G for 10 minutes, you get a supernatant. And then the clear layer, you don't want this is not what you want yet. What do? You pour out the clear layer, which is called the supernatant, and what you have under is just the nucleus and the unbroken cells, which are like the heaviest thing you don't need. So what do you do again? In centrifugal centrifugation, in differential centrifugation, you don't just do it once. You do it in increasing speed. Now look at the second step involves from 600 to 15,000 G. You spin it once again. Another time, what do? What are you going to spin? You're not spinning your residue. You're spinning the supernatant. So you pour the supernatant again, balance it up in a centrifuge and spin it again. After spinning it for 5 minutes in 15,000 G, what happens again? You get another clear layer. This clear layer now, and then a debris, the debris or the residue. The residue here now is the mitochondria, the lysosomes, and the microbodies is what you see. You get this supernatant, which is another clear layer, and spin it again for the third time. In this third time now, you increase the speed drastically. You are increasing the speed from 15,000 in this second step. Remember, this is 1. Now this is 2. In the third step, look at what you're increasing it to what? 100,000 G, and you are spinning it for 60 minutes. And after doing this, what do you do finally? You now get in the third step, the third clear layer will be the soluble fraction of the cytoplasmic content, including enzymes you may be looking for. And then this particular debris here contains the ribosomes, the microsomes, and then the radial reticulum, the Golgi apparatus, and all the rest of them. And it is this one now that you take and continue your analysis. So you continue with whatever you get from here for analysis. So this is what we call differential centrifugation. Differential. So you are increasing the centrifuge as you go down the steps. Now, sorting out. Sorting out is a purification test for protein based on the differential solubility in salt. Now, it usually depends on the ionic charge or the ionic strength or the polarity of that protein. Now, a good example salt used is ammonium sulfate, which usually what does it do? It takes away water from the cell. And when you take away this water from the cell, what happens to it? The cell begins to what? Precipitate. So it takes away water by interacting with it and makes the protein less soluble because hydrophobic interaction among proteins will increase. Now, so you can also take different aliquots and now taken as a function of the salt concentration and then get them closer to the desired protein sample. All right. So you can now get your fraction of interest from that place. And then we move to the next step, chromatography. Chromatography is an important technique that have gained a lot of attention and has actually contributed in purification of a lot of substances, not just proteins, purification and separation. So it's based on the fact that substances can distribute themselves in two layers. Those layers are the mobile phase and the stationary phase. Now, the stationary phase is the one that doesn't move. So what it simply do is that it retards the flow of substances in the what? Mobile phase. And in such a process, there will be separation. So if the substance has a lot of interaction with the stationary phase, it's not going to flow. What it means is that it's going to want to stay and allow the mobile phase to go through. So the mobile phase is the portion of the system in the mixture, is the portion of the system in which the mixture to be separated moves. It is that one that moves along with what you're going to be separated. And at the end of the day, you're going to now get your product by elution. So column chromatography is particularly used and has offered a lot of promise. Column chromatography is a form of chromatography in which the stationary phase is packed in a column. Here we are talking about a stationary phase that is packed in a column. What do we mean by column? Column means you're not using common paper chromatography now or the thin layer chromatography. It is a column. You have a column. This is what we mean by column. This is what we mean by a column. And here now, you pack your stationary phase. Your stationary phase can be a bead or anything, or a gel. And then you now pour your solvent. Your solvent, of course, will be the mobile phase. Of course, remember that. And then this is the stationary phase. The stationary phase can be a gel. And then you now put your mixture, your sample. Your sample can now go inside. So it has offered a lot of promise in purification of substances. So this is a very good example of a column chromatography, much more clearer than what I was drawing. So now in a column chromatography, you're going to have a column this way. Now, inside this column, you're going to have a packed stationary phase. This is your stationary phase, though this looks clear, but something is inside it. Now, this is a tank or a reservoir. Now, inside this place is where you place the sample. Now, what happens? The sample will begin to elute. So as the eluent flows through the column, compounds of the sample migrate in different areas. Now, look at what happens now. Depending on the relative attraction of the sample with this stationary phase, one of them might be more attracted at this point, the other one will be more attracted at this point, and as the mobile phase is going down, you see that these guys are separated depending on where they have the maximum attraction. This type of separation is what we call column technography. So what are you going to do? You can now begin to elute them or collect them individually. So what do? You open up the tap, you collect, you first of all try to remove what you don't have here, and you get here, you collect this one, you stop. You go to the next one, collect this one, you go to this one and collect this one. So the eluent is collected manually, automatically, and they're analyzed for the presence of what you are looking for. And in fact, this is a graphic way or a graphic principle of representing most column chromatography that are used today. Let's now begin to see those individual types of column chromatography. Size-Exclusion or Gel-Filtration chromatography. This technique is used to separate biomolecules based on size. The name is actually mentioned there, based on size. So the size matters in this case. So it separates based on size of the biomolecule. The stationary phase usually consists of a cross-linked gel, a gel that has different spaces, opening or pores. The extent of the cross-linking of this gel is controlled by the pore size. That's what controls the size of the pore. The extent to which this gel are cross-linking themselves will create. So if you have a gel, let's say, let me put it this way. I like trying to draw gels this way. That is cross-linked. You find out that this looks like a molecular sieve. See a sieve now. There are openings here. So one of the samples can easily go into this place and pass through. If they're too big, they can pass through. If they're very small, they can pass through and go through area. So this is a kind of molecular sieve. And one of those good examples used is the agarose gel. The agarose gel are made in different pores that can be used as a very good molecular sieve in gel filtration chromatography. So this is a very good example of a gel filtration chromatography. So in this case, you have a mixture of different molecules. So a mixture of molecules of different sizes. You now apply them into this gel. So the gel, like I said, is the stationary phase. Look at how the gel looks like. The gel has different pores, like the one I drew earlier. So it's a bead cross-linked in different areas. So what happens? When you pour them in, you look at the two samples we have here. We have the green one and we have the red one. When you pour them in, what happens? You find out that the red ones will stick inside because they are much more what? The red ones are much more bigger and they will hang inside the bead. They can't go through. Whereas what? The green ones will not even get inside the gel because they are too big to get inside the gel. What do? They just get eluted. So what you do is just to collect them in different times, and when you analyze them, you find out that they look at the elution profile. The elution profile shows you that this is where you have the maximum collection of the green ones, and this is the maximum collection of the small ones, because the small ones goes into the gels and they're stuck inside it because they can go through. Whereas the big ones cannot go through. What do? They just get eluted first. In those two ways, you can separate these two substances based on their molecular sizes. So you can also call this a molecular sieve chromatography. Affinity chromatography is even much more specific. In affinity chromatography, a column is used that contains a substance that a protein or enzyme can attract with. Affinity simply means, what is attraction? If something has an affinity for something, that means it has a strong specific attraction to it. So it's a powerful column separation procedure based on specific binding of molecules to a ligand. A good example, every enzyme has a specific substrate. So if you have a particular substrate in a column and you want to separate the enzyme of that, if you put it in there, the enzyme is going to bind only to that substrate, and then every other stuff is going to become a waste. And that is exactly what is demonstrated here. So this column with a substance S, the substance S here, look at the substance S, covalently linked, is covalently bonded to the supporting material here. So the sample containing a mixture of proteins are poured in. You see them containing a mixture of, so this is substance S. If you look at what happens here now, protein 1, we say P1 molecules will bind to S because it is specific for it. So look at this. The P1 molecule that is this red triangle will bind to S. Look at it. So it's just like when you have, we're going to see this. Okay, I'm going to change my color. When you have enzyme plus substrate, which is specific, they form enzyme substrate complex. That's exactly what is happening here. The protein here, the enzyme, as the case may be, binds to this substrate yellow, and they just form a close affinity. And then the rest of what you have is going to be removed as a waste. And what do? You can now adjust the pH of the buffer, and then you elute your what? Your proteins. And then you've been able to separate protein P1 from protein P2 and protein P3. If you look at that, at the first one, the protein P2 and P3 is the spherical and square. The spherical and the squares went out initially because you don't need them. But the P1, which looks like a triangle, sticks to each substrate and stays there and then can be collected lastly. So this is a powerful tool because it is highly specific in nature. the ion exchange chromatography. This is a method of separating substances on the basis of their net charge. Now, in this case, we are looking at the overall charge. Remember, when we talked about proteins and amino acids, remember, there are amino acids that are positively charged. A good example of an amino acid is lysine. A good one that is negatively charged is what? Is aspartate and glutamic acid. Now, there are two types of exchanger you can use. In cation exchanger, you use a resin or a stationary phase that is negatively charged. What that simply means, if you have a stationary phase that is negatively charged, it's going to bind to what? A protein that is positively charged. That's exactly what it is doing. So a stationary phase or a resin that is negatively charged will bind protein that is what? Positively charged. And that's what we call it a cation exchanger. And if you have a stationary phase or a resin that is what? Positively charged, is going to bind to a protein that is what? Negatively charged. We call it the anion exchanger. And that is exactly what we have in this. So in this cation exchanger, let's see what happens. Now, if you see what is happening in this cation exchanger, you find out that now a cation exchanger is going to have a net negative charge. Let's see. If you look at all these particles, this one is negative, 4 negative. This one has 2 positive and 4 negative is charged negative 2. So all these guys, if you look at them, count the number of negative and positive, they're all negatively charged. So a negatively charged resin or stationary phase is going to attract a positive protein. Let's look at the 3 proteins in here. If you look at these 3 proteins now, this one has a charge of 1 negative, negative, positive, positive. It has no charge. That's exactly what we're saying here. The same thing with this one. This one has negative, has what? Yeah, it has a negative, negative, and a negative, and it has a net charge of what? It has a net charge of what? Positive. That's what it means. It has a net charge of positive 1. So it has a net charge of what? When you say 3, this is 3 minus 1, it's going to give you negative 2. But that is not what we want. Remember, a cation exchanger will do what? A cation exchanger is going to attract proteins that are what? Positively charged. Positively charged. So if you look at these 3 proteins, the one that is positively charged is only this one. Because if you look at it, it has plus 1 here, plus 1, which is 3 plus. 3 minus 1 will give you what? 2 plus. So this is a positively charged. What that simply tells you is that this protein is the only one that is going to do what? That is going to be attracted to any of these bits. Or you can even say the one that is closest. I don't have to draw. This will be attracted to this. And these the other 2 will be eluted first. So let's see that. If you look at it, as they are moving, this one that is what? Plus 2 charge was attracted to this particular cation exchanger. And then the other 2 does what? Leaves immediately. And when they leave, you cannot change the buffer of this one by doing what? By adding more what? More positive charges to elute it at the end of the day. And you've done your separation. And this is what we call the ion exchange chromatography. So high performance liquid chromatography is also called high pressure liquid chromatography. We have this in our lab. It is the most sophisticated chromatography technique that gives a fast and clean purification. Now, in the reverse phase HPLC, you use a form of high performance liquid chromatography in which you use a stationary phase that it has both non-polar and the polar phase. And the good thing about the reverse phase is that you can separate both the polar and the non-polar substances at the same time. And the discovery of the HPLC has offered a lot of interest and a lot of promise in chromatographic techniques. So it uses high resolution columns that actually take place in a high pressure. In fact, at this time, most of the substances need to be vaporized. So you see, they need to be vaporized at high temperature. And at that place, this separation occurs. And it gives you a very clean and high resolution. So it uses a high resolution column that takes less time for separation than normal standard column. So high performance liquid chromatography has been staying around for some time, and it is a very good way of separating your proteins. All right. So electrophoresis. This is a method of separating molecules on the basis of their charge. And size as well. So let's say charge to size. So you look at the charge ratio to the size at the same time. That's what it does. Now, in this case, it looks like, I tell students, it looks like electrolysis, but in this case, it is no liquid. It is a gel. The stationary phase is a gel. A gel is applied onto a plate, and then a voltage is passed through it. Then This is the plate that has been applied, and then you spot what? You spot your samples all through this. What the samples does is to move and then get separated depending on the charge and the size. On their size, they get separated. So the best gel used are usually agarose gel is the best used for nucleic acid. We use the agarose gel for nucleic acid. The polyacrylamide gel is used mainly for proteins. And then it has more resistance towards larger molecules than smaller molecules. We're talking about the polyacrylamide. So this is a simple electrophoretic machine that you can see in most of your lab, particularly for biology students that will do that. You see this. So this, it has the two electrodes that will bring in the charge in between them. And inside this place, you have the plate. So this, the gel box, and then inside it is have the plate. So when you use it for separation, in SDS means polyacrylamide gel electrophoresis, or you say SDS page, you see that the technique separates protein based on their size. The protein is treated with this detergent, sodium SDS, we call it SDS, sodium dotyl sulfate, that's what we call it SDS, polyacrylamide gel electrophoresis. So smaller proteins are small according to their size. Remember, it is charged to size. Charge to size. The small proteins will move faster than what? The bigger ones. The bigger ones will be slower as they are being separated. So this is where the separation starts. You look at this, it moves along this way. The lower ones will move faster and get separated, while the smaller ones will be at the back. So electrophoretic technique is very good, particularly applied in separating nucleic acid. It's very, very common. The isoelectric focusing. Remember when we talked about this in protein? The isoelectric point is the pH of protein at which it has no charge. We will say it's the pH at when it is what? A sweeter ion. So it's a method of separating based on using this idea of this isoelectric point. A gel is prepared in a pH gradient that applies to the electric field. Now the charge on this protein changes as it migrates across that, as it migrates through the gel under the influence of electric field. Under the influence as it moves. Now if it gets to a point where it has no charge, remember when there's no charge anymore, there's no more attraction between the poles of the electric field, what happens? They stop migration. And you can use the idea of that PI, or what we call the isoelectric point, to begin to isolate and identify individual amino acid. Remember, we did this in proteins and in amino acid. Every amino acid has its own characteristic isoelectric point. And that, using the idea of that PI, you can differentiate different amino acid or proteins in a particular molecule. Now, determining the primary structure of protein. You see, determining the primary structure of protein has been a routine in the development and understanding novel substance biomolecules, particularly the proteins and the complex proteins that are now everyday research is going on for people to understand the various versatile functions of proteins. Now, so what do you want to determine? Remember, we said the primary structure of protein is built on the N-terminal part of the amino acid to the C-terminal part that are held together by what? The peptide bond. Remember, it is just the peptide bond that hold them together. So if you can establish the individual amino acid present in this molecule, that's what you want to do. You want to establish those amino acids. And you want to determine which amino acid is the N-terminal and which amino acid is the C-terminal. And then you can divide these proteins into smaller fragments in determining this sequence. You can say, okay, if it's the protein that is 50 amino acid, you can break it down into, let's say, 10. And you begin to look at, okay, which amino acid is the N-terminal of the first 10, which amino acid is the C-terminal of the first 10, you go to the second one. So this is what we call determining the primary protein structure. So that is exactly what has been shown here. So if this is a peptide that is going from normally N to C terminal, and you want to determine the true amino acid component of this, what do? You begin to first of all break them down. What are the things you can use in breaking them down? You can use some molecules, or even better, enzymes. Remember, enzymes can break this like peptidases can break this down. So you can use enzymes to break them down. And when you break them down, what do? You isolate this and begin to discover them. Now you know all the amino acids that are present in that protein. You can do this using either your chromatographic techniques or mass spectrometry or any other technique to begin to analyze this. And then step 2, what do? you can use specific reagents now to begin to do what? To begin to determine these ends. Use specific reagents that can react with nitrogen of the N-terminal and carboxylic acid of the C-terminal, because you want to know the identity of the amino acid that is at that particular point. In step 3, you can now cleave the proteins at what? Specific sites as well. You can break it down into the simpler peptides and then begin to determine what type of peptide bond. Remember, a peptide bond depends on the 2 amino acids that are in contact. And lastly, you can cleave the protein into specific other sites. Apart from this peptide, you begin to look for which amino acids are in those sites. So this is a typical example of how to do the primary structure of protein, or you can see determining the sequence of protein, because the primary structure of protein determines the amino acid sequence. So this is a very good example of what you can see. If you digest your protein with an enzyme or any chemical and break it down into the corresponding amino acid, you can use a HPLC to identify those amino acids. And so this is a good example of a chromatogram that contains the following amino acid. And if you do this, it is a positive step in knowing at least the identity of each of those amino acids in that particular protein. Now, determining the protein sequence, like I said, involves the cleavage of this protein by reagents that produce the mixture of this polypeptide. And this polypeptide can be separated by the HPLC or other chromatography techniques. It uses several reagents on different protein samples to be sequenced in different mixtures. This is exactly what happens here. So you look at the chromatography can be used in breaking this apart. You can also use cyanogen bromide. You can use chymotrypsins in each of these stages. And at the end of the day, you can determine the overall sequence of this protein. So it's actually a very complex process, but which has recently been replaced by a better equipment. Now, the man that gave us the greatest insight to the understanding of protein sequence is called Edman. And that's why we named this process after him, Edman degradation. What Edman did was to use enzymes. So he used the Edman method to determine amino acid sequence of peptides and proteins. In this method, you can sequence up to what? 10 to 40 amino acids in 30 minutes. The principle has been so effective. It has become so efficient that the identity of the N-terminal, amino acid at N, and the amino end, and the carboxylic terminal of protein have been very much determined from this process. However, this is just the principle. This process has been replaced with sophisticated instruments like HPLC, like mass spectrometry, that you can now use that offer the process even much more faster. But Edman has credit for letting us understand the principle of protein sequencing, or how to determine the primary structure. And of course, determining the primary structure of protein is an improvement, because the primary structure is what determines the what? The secondary is what determines the tertiary structure. And the tertiary structure is what tells us the native state, and it is the full native protein that can perform its biological functions. And this is a very good invention in understanding of the knowledge of biochemistry. Protein detection techniques also. Now, ELISA technique. This ELISA technique simply means enzyme-linked immunosorbent assay. Remember, in enzyme-linked immunosorbent assay, you believe that, now, a typical thing that happens to the body is that a foreign body that comes into the body is called an antigen. And every antigen, if the body needs to protect you, has an antibody that will attack it. An antibody. Now, in enzyme-linked immunosorbent assay, you see the immunosorbent, exploring the knowledge of antigen-antibody interaction. That is exactly what is happening here. The antigen-antibody interaction, which usually leads to agglutination or precipitation, is what is used. So here, an enzyme is only used to mark the antibody, because that's the only way you can mark the antibody. If you use enzyme, you mark the antibody, and the antibody is going to interact, and then you can use the activity of the enzyme and the substrate that it can process to determine the quantity of this particular protein. That is, so this picture shows you. Now this is the antibody. Ag, that's what we call the antibody. Now, sorry, this is the antigen. Sorry, Ag means antigen. The antigen is Ag. The antibody is AB. Now, the antigen interacts with this antibody because it's specific. This is highly specific for it, and it forms agglutination. Now, how do you measure that agglutination or precipitation? That can be done by telling you the activity of this enzyme, because the enzyme is used as a marker. That's what we call it, a linker or a marker, enzyme linked. The enzyme now, every enzyme is known by having a particular substrate in which it acts on. So if you can incubate this enzyme with a substrate and calculate the activity of this enzyme or how much substrate it can produce, that can be used in proportionally estimating the amount of what antibody anti-protein interaction that have taken place. This is what we call the ELISA technique, and it is pretty specific in nature. So finally, we're going to be talking about the last page of this class will be proteonomics. What is why do we even have to talk about the understanding and the sequence of protein? That the information cannot be overemphasized, because proteins run our body, the metabolic processes, the enzymes, the hormones, and everything. So if you're going to understand the pathology of a particular disease, you need to understand the proteins, the protein makeup of a particular enzyme. They may have been defective, and that could offer an idea on how to control it. So what is proteonomics in this slide? Proteonomics is the interaction among all proteins of the cell. That's what we call proteonomics. We want to study those interactions in all the cell. So in other words, you want to study the proteome. The proteome simply measures the total protein content of a cell, or the total protein makeup, including the enzymes, the hormones, the structural, every part of the protein that make up the cell. And those types we can look at are, one, the structural proteins. This is the detailed analysis of the structure of the protein being produced. You want to know the structure of this particular protein. What does it look like? What is the primary, secondary, or tertiary structure? That's what you look at in this. In expression, you are analyzing the expression of this protein and consider the expression under different conditions. Now, how will this protein be expressed in different states? And how does it have to interact with the genetic code? How will the genetic code determine the expression of this particular protein? And lastly, you want to determine the interaction. The interaction offers the opportunity to look at how this protein interacts with other molecules in the body. And this is what we call the proteome. So it's very interesting because proteins are so versatile, both in structure and in function. There is need for scientists to keep studying and understanding their nature. And this will keep offering great insights on how to do what? Understand both the pathology and possible cure of many diseases. Thank you once again for listening. We're going to stop at this point. Enjoy your day and bye at this point.